inflammation and on decreasing inflammation by these signaling, manipulating signaling pathways. Now, what cells do, they basically integrate different signals. Like our brain, when it hears different sounds, integrates these sounds into sequences that are understood by us as speech, as language. This is what the cells do with the signals that they receive. So the proteins in each cell receive a complex combination of signals which simultaneously trigger many different signaling pathways which provide an opportunity for crosstalk between different signals. Now, signaling includes different cell-to-cell communications, and this slide shows how basically communications happen. And all these light up areas are basically signals or communications that are happening at that time. Now, there are different functions that the cells fulfill by these signals. So one is extracellular matrix communication, and this is, you can see here how this can happen. All these lines that combine these two cells are sort of a communication network that is formed, just like the internet very much, or other communication media. Now, also, the cells are leading to different cellular responses, which one is adhesion, another one is migration, essential for tissue formation in wound sealing. So different cells migrate in another area to kind of heal the wounds. Proliferation, this is how cells increase, basically, and differentiation. Now, you cannot have proliferation without differentiation. It won't work. Proliferation without differentiation is going to give you a tumor. It's going to give you a cancer. So that's what cancers are. They just proliferate, but they don't differentiate, not like the normal cells do. They do have a cellular differentiation, but not quite. Now, what differentiation means is that after the cells proliferate, they become different cells. They become a muscle, for example, they become blood, they become a nerve cell. So this is the cells prior to differentiation, and this is the cells after differentiation. So, but the problem is that the greater the complexity, the larger the possibility of communication mistakes. And the way we understand today aging and illness is that it's nothing more, nothing less, but a communication mistake. So clusters of multiple ligands and signaling receptors interact with each other, and specific cellular responses result from the integration of multiple incoming signals. And any aberrant communication between cells and their surroundings, extracellular matrices, can lead to a plethora of human diseases. And for example, sclerosis, arthritis, renal lac fibrosis, and of course cancer. Aberrant communication between cells and the surroundings and extracellular matrices also leads to aging. Now, signaling, how is signaling decoded? How is there a way of decoding signaling? Yes, there is. The way we do it is signaling decoding is relied on simultaneously crystallizing and measuring multiple proteins in thousands of individual cells. So you do that at the same time. And this is how you look at signals. That's how you decode the signals of the cells. Now, perturbing these cells with molecular interventions drives the ordering of connections between pathway components. So different cells you take, is the way we speak. Just always keep in mind the frequencies of spoken language and how these are combined into words. So it's just sounds. You combine this into words, and these words have a meaning eventually. It's the same thing. It's exactly the same thing. It's like a complex computational process. And to decode that is another very complex computational method that automatically looks and kind of decodes the signaling relationships. Now, reconstruction of network models from physiologically relevant primary single cells can be now applied to amplify faded configurations of signaling pathways to ultimately replace dysfunctional signaling in aged or diseased cells. How is signaling decoded? There's more. Another body of researchers is exploring the resonance that is mediated. Basically, as the cells communicate, there is a sort of a resonance. If the two of them click, there is a resonance that happens. That means resonance is amplification. That's what resonance is. So resonance-mediated increase of signals is involved in the synthesis of certain proteins, such as collagens, elastin, hemoglobin, actin, myosin, adipase, that's the energy that forms the ATP, hormone secretions such as DHEA, that is for the skin and the bone, T3, that's for lipolysis, and IGF-1, that is the hormone that builds the muscle, responsible for the muscle. Now, what is the purpose of decoding cellular signaling? What we want is we're trying to form a universal language of our cellular microcosmos, like a new language, the language of life. And here is how you can see like an insulin code. This is the code of the insulin. As you can see, it starts with a sequence of ADG, and it ends in AGG, and then there is another sequence and which ends in ACC. This is like letters, and basically this is a code. It's how you create, this is how you would write it, for example. But in fact, it's a conglomeration of different frequencies. Now, missing signals can leave the cell with incoherent instructions, and therefore they're going to give inappropriate responses, and that will be deleterious to the cell. This is what another theory of cancer is, that it's the wrong signaling. Now, investigating how such signaling pathways are organized is relevant not only for understanding how normal cells work, but also for appreciating the molecular basis for aging and disease, since aging, as well as many human disorders, result from breakdowns in signal transduction. Now, this is a research that was done on cells, and it was done on the basic research by Xanya et al. That aging is the result of protein communications that have diminished. So here, out of 4,000 different proteins that were identified, these proteins comprised about 7,103 protein-to-protein interactions, almost twice as many. And the way it's done, it's not in a sort of a linear fashion, but it's sort of in a very complex way, where one protein gives signals to many other different proteins. So what you see is that the result of examining all of these interactions, the 7,103 protein-to-protein interactions in aged versus young subjects, what this demonstrated is that aging is the result of the disorganization in this interaction. So the more disorganized these interactions are, the more there is aging. Now, in Cambridge, the research looked at signaling properties of young versus aged cells, and then tried to sort of transfer the signals from the young cells into the aged cells, and what they found is sort of a rejuvenation, if you like, of the aged cells. Now, ion channels are crucial in signaling. Ion channels are also intricately related to a number of biological functions. For example, calcium signaling has been proved to be strongly associated with aging, manic depressive illness, and neurodegeneration, or optimal neurological functioning. And in stem cells, what we have found, very interesting part, is that when the potassium channels are blocked, and this is a Johns Hopkins University research. What you see is that you see no differentiation in stem cells. And if stem cells do not differentiate, like I said before, they will form tumors and then cancers. So potassium channels are crucial in stem cell differentiation and absolutely crucial in stem cell therapies. Now, another very interesting fact that came out from recent research is that the electrons are actually controlling ion channels, but they only control ion channels at very extremely low frequencies. So all of these electrical machines with microcurrent, they're all wrong because they cannot control, they cannot do much about signaling pathways. In order to control signaling pathways, you have to have something that is extremely minuscule, like nanocurrent. So at those energies, then you can have ion channels being controlled by electrons, and you have something kind of to work with. Now, so you need that, and then you need, of course, a matrix of signals that are connected to form the language sequences of biological functions. And then you can try to do that by creating sort of a meaningful pattern, or the way verbal speech is produced. Now, there is some research that about wound healing, and it has to do with certain collagen receptors. And what they found is that DDR1, you did see DDR1 and DDR2 right here. This is DDR1, DDR2, okay? And this is the functions that go with it. And what they found is that DDR1 is not essential for wound healing. It's linked to excessive collagen deposition during keloid formation, but it's not good for wound healing. So it makes the keloids and the scars, but it doesn't do much for the wound healing. Now it's DDR2 collagen receptors that are therapeutic targets. So again, you're looking at signaling, you see? Because it's what you stimulate, it's what you trigger that makes the whole difference between perfect healing. And this is probably a very important kind of information for eventual research. Now, we've done a number of clinical studies, and the way we did it is we used judges to evaluate the results of before and after pictures. And treatment was done either one side of the face, or we had some wound healing pictures, and we also did treatment both sides of the face. And all after pictures, or the after picture that we took were several months later to see if the results were going to be lasting. Results were really unambiguous, so the agreement between the judges was extremely high. So we had a very high correlation of very significant results. And this is just some of the pictures of the wound healing. This is a traffic accident, and what you see there is pretty much the bone. This was very deep, and this person went to a plastic surgeon. The plastic surgeon, how you will never grow nails, that's it. But he did after, and he healed quite well. And this is a recent, this is after 4 treatments, and this is the before, this is the after 4 treatments, which were done like in about a week. And you can see there's quite significant healing there as well. This is just some of the pictures that you can see in our booth, the before and after that we used. Some more, that's just one side of the face. You can see the no wrinkles. That's something you can actually see in the booth on your own faces very easily. That's another before, half side down, half side is not down. This is a full face down. This is courtesy from